Thank you for joining us this afternoon. My name is Trey Foskett. I'm one of the co-founders and CEO at Watchmaker Genomics. My background's been in life science tools with a focus in protein engineering and next-gen sequencing. Today I'm going to provide a brief overview of Watchmaker, some of our core capabilities, and then how we've been applying protein engineering and application development with some examples in sequencing, and then I'll hand it over to my colleagues. All right, so I always like to start with this slide. We're a mission-driven company, really excited about the positive feedback loops that come from new technologies. So that intersection of biology, engineering, and data really creates so much of the innovation that ultimately gets expressed and impacted by a lot of folks in this room and at this conference, having a real impact on basic science and research as well as human health. So we're really motivated by this mission as a team. So Watchmaker products are used to power precision genetic testing. So our customers range from centralized, ultra-high throughput LDT sequencing labs all the way through to point-of-care field-based diagnostics. Our products are used in NGS, PCR, qPCR technologies, and they're there to enable applications such as liquid biopsy for oncology, as well as infectious disease testing. And so feel really fortunate. We get to collaborate with some of the most innovative assay developers and sequencing and diagnostic companies in the world. We've got an amazing team. We're growing very quickly. We're about 225 full-time employees. And from the very beginning, we've been really focused on building a strong culture of quality. So we've been ISO 13485 certified for several years now, and that's really established a lot of confidence and trust with our partners. We're headquartered and we manufacture in Boulder, Colorado. We're supported by a global team in Europe, China, and South Africa, and we're vertically integrated, which means we control from product design, process development, through manufacturing, all those steps in the process, and we can really control our quality. We have very fast turnaround times, flexibility, and scalability embedded into our operations. So our foundational strengths are in protein engineering, in computational biology, and in large-scale protein manufacturing. However, I really feel what defines us as a company is our focus on these capabilities toward identifying bottlenecks in molecular diagnostic applications where better enzymes and reagents translate into better data quality, workflow efficiencies, and ultimately improvements to assay sensitivity and specificity. So we're very applications focused as an organization. And core to many of our products are differentiated DNA and RNA modifying enzymes. Those enzymes then get incorporated into application-specific kits and formulations. And we've got a product development process that leverages a few different strategies in protein engineering. So rational design, computational design, directed evolution. We then couple those with microfluidic formats and deep sequencing readouts so that we can characterize and screen, in some cases, millions of enzyme variants in parallel. These methods generate massive amounts of genotypic and phenotypic data that we then use to quantify subtle changes to protein structure and function. We then feed that data into our computational models that then inform us around the next steps in our engineering and product development process. So this is an iterative process where we improve every new enzyme and product we develop. We get better and our process gets better. So this framework around product ideation, high throughput screening, deep characterization, gives us a lot of control and precision in how we think about engineering enzymes with specific characteristics that are relevant for molecular diagnostics. So for example, improvements to catalytic rate and speed, inhibitor tolerance, thermostability, et cetera. These are all properties that improve the robustness and sensitivity of molecular assays. So as I mentioned, we use a couple different methods in protein engineering. Historically, these have been more siloed, and it's been really exciting over the last several years to watch these various methods become more integrated and more synergistic with each other. Starting with rational design, that's a situation where a human takes protein structure function information and makes inferences about what amino acids may change to confer some improvements to the protein of interest. Computational design where algorithm can take protein structure, function information, and make predictions around various beneficial changes and mutations that could be made in the protein of interest. And then deep mutational scanning and directed evolution, they're conceptually related concepts insofar as they're both hypothesis-free, meaning no a priori assumptions of structure and function are required around the protein of interest, and they both require a lot of genetic diversity. So you tend to create very large gene libraries of enzyme variants. Directed evolution uses an iterative process of mutation and selection sequentially, whereas deep mutational scanning leverages the power of next-gen sequencing to measure thousands of mutations and the impact that those mutations may be having in parallel. So in the next few slides, I'm going to use a reverse transcriptase as a case study to walk you through a little bit about how we use some of these tools in protein engineering with a particular focus in deep mutational scanning. I think I've spent most of my career trapping enzymes in bubbles. And so this is a pretty simple schematic here around a microfluidic format that we use to give us more control, more precision over the various reaction parameters of an experiment. So what we'll typically do is take a gene of interest, in this case a reverse transcriptase, express it, transform it into E. coli, express it, and then encapsulate the cells into droplets. And so effectively each droplet becomes a microreactor in an experiment of interest. And so you can see here at the bottom, cells will flow in, reaction components will flow in from the top, they'll be merged into an aqueous phase, and then that aqueous phase is then merged into an oil phase that promotes and maintains compartmentalization. So there was a method called compartmentalized self-replication that was pioneered, or CSR, that was pioneered by Phil Holliger and Farid Gadesi at the MRC over 20 years ago. That method was very specific for thermostable DNA polymerase evolution and engineering. We've adapted that system for reverse transcriptases and other mesophilic enzymes. And essentially what's happening here in this process is called compartmentalized self-reverse transcription. And I've already walked you through on that previous slide, we'll take the gene library, transform it into E. coli, encapsulate the E. coli into droplets, and then here on the second step, once the E. coli cells are lysed, you have two scenarios. The scenario on the left is an example of a reverse transcriptase variant that is active from the library, and it's able to reverse transcribe its RNA into cDNA, and therefore enriches its genetic information. And then on the right, this scenario, I do have a pointer, here is an example of an RT variant from the library that was either inactive or not able to survive whatever selection, pressure, reaction conditions we provided it, and it's not able to reverse transcribe its RNA into cDNA, so therefore its genetic information is not represented in the population. So this is an elegant strategy where we're able to effectively recreate the survival of the fittest at the molecular level, and we can apply various different reaction conditions and selection pressures on the system. So for reverse transcriptases, for example, we've done improvements of thermostability, template switching, inhibitor tolerance. I'm going to talk a little bit more about template switching next. Okay, so in addition to the functional screens, we pair those with deep mutational scanning using NGS, where every single amino acid substitution, we can quantify its impact on enzyme performance. So this is, if you haven't been listening, this is the one slide that I think is most exciting for us. So on the x-axis here is the protein sequence, about 750 amino acids for wild-type reverse transcriptase. On the y-axis here is a fitness score, a relative fitness score. And then in the different colors here within each amino acid residue is the amino acid substitution at that position. And so what you can see immediately is that most mutation when it's introduced into a protein, is deleterious. So then this represents, negative decreased fitness of that enzyme. So those are typically conserved motifs or the active site or areas that the protein doesn't like to be modified. But you can see above the baseline of the wild type hotspots, areas where clearly there are changes at the, with various amino acids that are conferring improvements to fitness. And so in this example, we were specifically engineering for improvements to template switching activity, which is a terminal transferase activity of reverse transcriptase that's really important in single-cell applications. We can also parallelize multiple selections or screens in various conditions. And so we were able to identify areas of the protein where we were improving the template switching activity. And what's really cool about the data presented in this fitness map is that we were able to recapitulate decades of research in MMLV, reverse transcriptases, for known mutations that were found in the literature, while at the same time discovering hundreds of novel beneficial mutations, all within a single experiment. So if you zoom in here on the fitness plot, you look up at the heat map above that. The previous slide, this was from a single site variant library. And then these heat maps in green are identifying the amino acid residue that conferred a positive fitness change. We're then able to combine those single variations into combinatorial libraries that then can undergo additional screening and selection, or we can combine those beneficial mutations into single reverse transcriptases of interest and do further characterization. And so my colleagues are going to talk more about inhibitor tolerance and thermostability, but this improved template switching activity we think will have significant impact on some of the single cell workflows, as well as long-read cDNA sequencing applications like isoseq. So a couple months ago in September, we made an announcement. So it's sort of warm off the press, an announcement with Promega, where we announced a partnership where Promega has licensed one of these engineered reverse transcriptases that will be incorporated into various, molecular diagnostic portfolio for Promega. And we were really excited about this partnership. We also think it validates our approach and the utility of some of these novel enzymes that we've been producing using our various technologies. And then we're really excited about some upcoming announcement with some additional partnerships that we'll be making available in the next few months.